This is a summary on the study conducted by Montgomery et al. Year 2007 entitled Differential VAR gene expression in the organs of patients dying of falciparum malaria. Plasmodium falciparum is a deadly parasite that causes malaria. The thing about these parasites is that they possess cytoadherence, making them hard to eliminate from the body. However, there's a way to locate them. The antigen PFEMP1 have adhesive domains that react to particular endothelial receptors. With this, a study on fatal pediatric malaria was carried out to find where Plasmodium falciparum infected erythrocytes are distributed through genotyping the merozoite surface protein 1 and 2 allele.
With the approval of ethical committees and consent from parents and guardians, this study chose 6 cases diagnosed with fatal malaria and had high blood parasite content from 3 different malaria seasons. Snap frozen organ samples undergo centrifugation and incubation before the extraction, and extracted RNA was treated for DNA contamination. 2 microliters of RNA proteins were used to make multiple copies of cDNA, which was used to make VAR clones. And lastly, for sequence analysis, Clostal W was used to match the sequences. PlasmoDB was used to search for sequence matches against the P. falciparum 3D7 genome. Peter Boll's database was used to analyze DBL motifs, and these highlighted codes are the accession numbers for the data sequence submitted to the following databases.
Before anything else, researchers first conducted a pilot study on the 6 cases of fatal falciparum malaria to know the diversity of VAR transcripts expressed by these parasites in patients' organs. Each organ had 20 VAR clones sequenced, so there was a lot of variation with up to 14 distinct VAR sequences discovered from a single organ. Thus, the researchers concluded that the number of clones analyzed was insufficient. So, to eliminate potential bias, the research design was revised. In this present study, researchers identified 3 patients with 3 different cases of fatal pediatric malaria. DBL1 alpha sequences were amplified from the brain, heart, lung, and spleen of the 3 patients. Each organ of the 3 patients had a median of 26 distinct VAR types. 1 to 2 are expressed in high quantities in the brain and heart. Mature PRBC were not found in PM55 lung, heart, or spleen, but they were found in large number in the brain.
A subset of VAR types are expressed at high levels in the brain and heart. Findings shows that there is less diversity in the brain and heart but are expressed at high levels compared to the VAR types detected in the lungs and spleen, but are more diverse. In most samples for every organ, there are 1 to 2 VAR types detected at far higher level compared to the other types. All these findings supports the hypothesis that the site of cytoadherence of Plasmodium falciparum depends on the VAR type, and suggests that the sequestration of this be organ-specific. Organ-specific VAR expression is observed in multiple patients from a single malaria season. The overlapping of VAR types between patients who were admitted months to years from each other, and some are from different villages, was not expected. This is evident between PM30 and PM32. The 2 most highly transcribed VAR types in the brains of PM30 and PM32 were also detected in PM36, an additional case from the same season. There are 3 additional cases to this study, and also displayed 15% of shared VAR types with each other and the 3 main patients. It was also found out that the VAR types organ localization were conserved, drawing a hypothesis of the potential of PFEMP1 as a vaccine candidate due to its antigenically variant nature. Using this molecule as a vaccine in malaria and pregnancy appears to be more promising in view of the very restricted and conserved nature of the PFEMP1 types involved in placental sequestration. This sharing of expressed VAR types between patients suggests that some form of organ-specific VAR expression, such as seen in placental malaria, may also occur in severe pediatric malaria.
It was discovered that VAR sequences did not have clustering with any of the 4 host organs, and that most structures had 4 cysteine structures with 12% VAR types containing 2 cysteine residues, with the exception of PM32, which had a sequence on their spleen that had 2 cysteine structures instead of 4 cysteine structures. They also looked at differential distribution between previous Duffy binding-like alpha motifs of positions of limited variance, but they discovered that this had no association with the site of sequestration, and it had a negative association with the antibody variant, and immune regulation of VAR types can be prevented from being expressed for pediatric infections. Many of the VAR types expressed in the heart tissue for PM30 and PM32 were eerily similar to 3D7 VAR types, which is the genome sequence for the human malaria parasite. Many of the VAR types expressed in the heart tissue of PM30 and PM32 were highly identical to 3D7 VAR types, which is the genome sequence for human malaria. All 11 VAR types detected in the heart of PM30 had a similarity of 70% with 3D7 genome, and 8 were greater than 80% identical. In the heart of PM32, 7 of 25 multiple copy phenotypes displayed high similarity to 3D7 bar types. Only one exception of these, and this was expressed at the heart of PM32. There was only a limited number of 3D7 genes to which the Malawian isolate showed similarity.
Overall, the results support the claim that accumulation of P. falciparum variant type is organ-specific and that PRBCs shows preferential binding in the body. And lastly, results on the overlapping variant types in the brains of children that died of the same malaria within a season suggest that there can possibly be a limited number of variant types that can sequester in the brain.